Good evening to all. Today I will talk about pet and human food, two parallel paths converging towards the onset of adverse food reactions. So I would like to briefly start with a short review of what are considered adverse food reactions. So they can be divided in toxic and non-toxic. The toxic can be can be food poisoning, such as microbial contamination, or non-toxic. Non-toxic can be subdivided in non-immune mediated, such as food intolerances, and immune mediated, IgE mediated and non-IgE mediated. Further, non-immune mediated can be subdivided in enzymatic, everybody knows the lactose deficiency, pharmacological, which is ascribed to the presence of vasoactive amines, which are such as histamine, which are responsible for some syndromes such as the Desgombroid 1. And also there is the undefined food intolerance, I would say food intolerance of idiopathic origin, and in this we hypothesize to be to place one of the most and widely present contaminant, such as the oxytetracycline. Oxytetracycline is widely and legally used all over the world, especially in zootechnic, and in particular for intensive farming of poultry, pig, and cattle farming. This is a paper of 2014 where for the first time they were summarized the minimal residual limits for oxytetracycline and other antibiotics, and where they were graded and evaluated according to WHO and FDA claims. So there is a difference between WHO and FDA of 1 to 10. So it means there is a huge difference of the minimal residual limits allowed for each of these protein sources. And this can give you an idea of what is the extent of this phenomenon. So our question is, there might be a potential risk to develop food intolerances of the entry or accumulation in pet and human food. I would say yes. This is one of our first clinical papers that we published in 2014, where we evaluated the presence of oxytetracycline and doxycycline. Doxycycline is used in poultry in pig intensive farming, and all these subjects, which were gym-trained subjects, were used to eat a lot of white meat, in particular chicken, and after 6 months of daily ingestion of such protein, they developed several food intolerances towards many other proteins, such as pork, chicken, soybean, egg, and so on. And having a look, analyzing if there was the presence of oxytetracycline and doxycycline within their sera, we observed that there was a huge presence of both antibiotics in most of the subjects. In particular, you can observe where there is an asterisk close to the value, and this confirmed us for the first time that there was a direct link between the daily intake of oxytetracycline food contaminated and the onset of food intolerance. This was a second paper that was published 1 year later, and was mainly an in vitro study where we evaluated the possible pro-apoptotic role of this antibiotic oxytetracycline in both in K562, which is an erythroleucemic cell line, and in PBMC, which are peripheral blood mononuclear cells from human donors, from healthy human donors. As you can see, there was a strong increase in the apoptosis percentage, after there was a strong increase in apoptosis percentage, in particular in a medium which was diluted 1:2 to 1:8 from the initial condition. And to give you an idea of the entity of this phenomenon, we have on the right side of these two graphs the positive control, which is represented by the oxytetracycline alone, or the hydrogen peroxide, which of course is known to induce a strong apoptosis phenomenon. But which was the procedure that we used? So we collected PBMC from peripheral blood of healthy human donors, and we took 1 gram of bone derived from chicken which were raised only with oxytetracycline according to the low limits. What we did was to co-incubate the 1 gram of bone of chicken with 10 milliliter of a medium for 48 hours. Then we filtered with a 0.20 micrometer syringe and diluted from 1:2 to 1:4 to 1:8. And then we put together the medium along with PBMC. And then we evaluated the apoptosis. This was a further paper, in vitro paper, that we published after, as a consequence of what we observed before. As you can see, we observed that after 12 hours of incubation with of a conditioned medium with oxytetracycline and PBMC, there was not a significant increase of apoptosis percentage, while after 48 hours of incubation there was a strong and significant increase of apoptosis percentage. Also, we evaluated the pro-inflammatory effect of this co-incubation, and only after 10 hours of incubation we realized there was a strong release of interferon gamma, which is a pro-inflammatory cytokine, either from CD4, CD8 and NK cells. So it had a very strong impact on the inflammatory background. And we also evaluated if OTC might be able to induce some the expression of interferon gamma and superoxide dismutase 1 mRNA. And it is possible to see here that in the case of interferon gamma, there was a strong and significant increase in the expression after 24 hours of incubation. We are talking about always about PBMCs. And also for SOD1, we observed a significant increase in the expression already after 12 hours of incubation. A possible explanation of this phenomena can be one that consider the superoxide dismutase 1 as an housekeeping gene, and its basal expression is usually higher than that of the interferon gamma gene. We also evaluated the presence of genotoxic markers after PBMC incubation with OTC. And the role, we evaluated the role of phosphorylated form of ATM and P53 and DNMT1. So the phosphorylated form of ATM is, which is a serine threonine kinase, is activated in response to DNA double-strand break to promote cell cycle arrest. And what we observed that there was a significant expression after 6 hours of drug exposure. As to DNMT1, it is an enzyme which is recruited to the chromatin in response to the oxidative damage, DNA damage, in order to inhibit gene transcription and to support DNA repair. As you can see, there is a similar kinetic with the p53, and both were significantly increased after 12 hours of drug exposure. A recent paper from Mueller et al. Reviewed and suggested some food allergens that are known to mostly contribute to the onset of cutaneous adverse food reactions in dogs, and this paves the way for our further slides. So they observed that the most present proteins were beef, fish, and chicken, and this also can it is easy to correlate what they observed with what we observed with our gene-trained subjects in our previous slide. This allowed us to perform a 1-year study where we provide new insights in the idiopathic food intolerances onset in dogs with a particular focus on the role of oxytetracycline and its metabolites as a possible triggering factors. So we recruited 8 client-owned indoor-housed neutered dogs and evaluated them for the presence of otitis, diarrhea, dermatitis, and generalized anxiety. For each of them, we analyzed the biochemical parameters, OTC serum, and kibble composition. Kibble composition, we are talking about the diet they were used to eat before displaying their symptoms. The biochemical profile didn't show any significant variation, and most of parameters were in the normal range. This after 15 days of organic diet supplementation to try to relieve their symptoms. And what we observed, and was in agreement with what we previously published, was a higher presence, a huge presence of oxytetracycline within the sera of these dogs in the order of 0.2 microgram per ml. And after 15 days of organic and oxytetracycline-free diet supplementation, there was a significant decrease in the sera of this animal. We also evaluated the kibbles of the diet which these dogs were used to eat before the onset of their symptoms. And it is possible to see here particle A and particle B, which are whose spectrum clearly show the presence of phosphorus and calcium, which are obviously ascribed to the presence of bone, which is one of the most constituents of kibbles, and also the presence of aluminum and silicium. So this is very important because there are many literature reports evidencing the pro-inflammatory role of silicium and aluminum nanoparticles, and they're also triggering activity in the context of the cancer. This was a mass spectrometry analysis which revealed the only presence of oxytetracycline within the kibble of these dogs. And this was also a great achievement for our research, which clearly demonstrated for the first time that there might be a strict correlation between the presence of OTC and the onset of chronic or acute inflammatory phenomena such as otitis, dermatitis, and so on. This is just some picture to show you which was the improvement of dogs. In the first two, there is a case of dermatitis, and before and after, it is clearly visible the improvement of the dog, and also concerning the otitis, and before and after the organic diet supplementation. We also, coming back to the cutaneous food adverse food reaction, we observed, we try to translate what we observed in dogs also in cats. And we recruited many, we recruited 18 cats, obviously client-owned cats, which were suffering from neck intense itch, neck eczema, and back and neck drooling. And we also evaluated the presence of oxytetracycline within their sera, and trying to possibly observe, as we observed previously in dogs, the disappearance of the symptoms, just providing an oxytetracycline-free diet. Here is a cytological evaluation where you can see microscopic images, A, ascribed to gingival swab with giant intermediate squamous epithelial cells with pink colored abundant cytoplasm, and where oral bacterial strains can adhere. As to the C square, it is an ear swab with papyrus-like enrolled elongated lanceolate corneocyte. And for the last square, we are talking about a derma swab with squamous epithelial cells with the pycnotic nucleus. You can see also the arrows which are present in the picture, and a papyrus-like elongated lanceolate corneocyte. So, as I told you before, we recruited 18 client-owned indoor-housed neutered cats, and divided them in two groups, one which received a standard diet and one which received a nutraceutical diet which was also free of oxytetracycline. So, and then we evaluated all their symptoms. I remember you back and neck intense itch, neck eczema, and also dermal lesions, and the presence of also oxytetracycline within their sera at time 0, after 20 days, and after 60 days. This was the severity and the extent of each lesion was scored according to a numerical rated scale with 5 degrees of intensity for the pruritus evaluation and 4 degrees of intensity for just for lesion evaluation, where we give a value of 0 for the lack of lesions, 1 for the very mild presence of lesions, 2 for a mild presence of lesions, 3 for moderate, and 4 for severe presence of lesions. As to the pruritus evaluations, we also used a scale from 0 to 4, where 0 was a condition where the cat was comfortable, grooming like a normal cat, and 4 as a condition where the cat was uncomfortable and grooming all the time. And this is a representative evaluation. On the left side, you can see the OTC concentration before and after 60 days of evaluation, just to give you a very direct impression of the phenomena. And you can see a significant decrease of oxytetracycline concentration in the nutraceutical treated group, and also a significant reduction of pruritus and skin lesions after always 60 days of nutraceutical diet supplementation. Our hypothesis is oxytetracycline, which is known to have an high affinity for calcium, which is obviously the main constituent constituent of bones, can remain fixed for extended period in treated animal, even respecting withdrawal times. And this was a published paper that we published in 2015. And although FDA and WHO established a maximum residues limit in liver, muscles, kidney, and fat, they did not provide any MRL for bone. Why? Because it is not considered edible. But although unfortunately, pet food production mainly relies on poultry byproducts, in which the percentage of bone meal is from 20 to 30%. So this makes it is obvious very often can drag behind the oxytetracycline residues, which are present in diet and can accumulate within pet's body, but also human, because bone is also present in sausages, in particular wurstel. We can conclude by saying that oxytetracycline represents the main employed antibiotic in zootechnical and veterinary practice as a food supplementation to ensure the wellness of intensive farmed animals such as poultry, swine, ovine, and bovine. We also showed that oxytetracycline is known to induce apoptosis in K562 cells and PBMCs. We also demonstrated the pro-inflammatory role of oxytetracycline. We provided data which partially suggested a causative relation between some toxic compounds, including obviously oxytetracycline residues, and the impressive clinical cases, the number of clinical cases which are daily observed in pet population. Our data also revealed an unexpected biological in vitro activity of OTC, which was also able to modify DNA in chromatin in cultured PBMC, and thus representing a potential risk for human and also animal health. Last concept is that our studies provided a clinical evidence of the presence of a plethora of symptoms which may occur in dogs fed on a commercially available, dogs but also cats, fed on a commercially available pet food diets, which have been shown to carry a huge amount of oxytetracycline. Nevertheless, we cannot rule out the possible combined pro-inflammatory activity exerted by nanoparticles and the presence of this antibiotic. Thanks for your attention.